Thank you all very much for coming to the seminar today. I am honored to introduce Dr. Andrea Glassmire for our seminar series today. It has been wonderful hosting her on our campus. If you had the chance to meet with her today, I'm sure you agree that her energy and candor for discussing science and life is infectious. Andrea did her undergraduate at the University of Pittsburgh before doing her PhD with Lee Dyer at the University of Nevada, Reno, studying the phytochemical diversity of tropical plants in the genus Piper. As a postdoc, she transitioned to more applied work with Will Wetzel at Michigan State, working on phytochemical diversity at different scales in agricultural systems. And then, with James Cronin at Louisiana State University in Baton Rouge, she applied her phytochemical expertise to wetland restoration research, which she will tell us a little bit more about today. Most recently, Andrea was hired as an assistant professor at the Plant Resilience Institute at Michigan State University. Yay!
Go green!
Where she has started her lab and which, as she told us at lunch, they just collected their first data. Yay! I have been a fan of Andrea's work for many years, and our paths have almost crossed several times through mutual research interests, collaborators, and mentors. So I'm so excited that she is here today to present her talk for us, entitled Phytochemical Diversity Regulates Resiliency to Herbivory and Environmental Stressors. Please join me in welcoming Andrea.
Thank you. That was so nice. That was like one of the nicest introductions I've ever gotten. I want to take that with me. I said go green, but I wore blue for y'all. Why can't we all get along? We're all doing really awesome science. That's what I learned today. I really want to collaborate with honestly everyone. So anyways, look me up if you're at Michigan State or you want to do something at Kellogg. I would love that after this talk. So what excites me about science is biodiversity and the mechanisms that maintain that diversity. And a lot of times when people think about diversity, we think of species like richness, or we think about different traits of diversity and sometimes even genes. But one component that tends to be overlooked is phytochemical diversity. And phytochemical diversity can really shape interactions with herbivores, predators, pollinators, and even microbes. And so, but its ecological role still remains unknown, a lot of the times because there's so many traits that are compressed right into phytochemical diversity. So today I want to show you what I think of when I think of phytochemical variation and its role in ecological interactions. And I'm going to show you projects spanning from tropical systems to, from tropical systems to agro-ecosystems and also coastal wetlands. And the overarching question in my research program is, how does chemical diversity structure ecological communities, inform pest management, and influence biological invasions? Okay, I don't have a pointer, but so again, when ecologists tend to think of diversity, they think of species diversity. And this is just a representative of the diversity in our world. So just frogs, for example, this is a small subset of many kinds of different frogs, birds, microbes, and fish. But chemistry is actually a critical yet hidden component within these systems. For example, this poison dart frog, I want to keep going like this, so don't mind me if I keep doing that. For example, the poison dart frog, I'm sure many of you know, sequesters toxins in its skin to help evade predators, and that's helped them speciate. But there's also birds such as the little shrike thrush that also sequester toxins in its feather to evade predators and microbes. Microbes are one of the key instigators for creating chemical diversity within plants and soils and other organisms. And then finally, there's this really cool chemical cue that is the basis between the mutualism of coral reef and goby fish. So when the coral reef are being encroached by the seaweed, the coral reef will send out this cue into the water, and it tracks the goby fish to come in and essentially graze the seaweed. And so chemistry is really an important component of these systems and many more. When I think of chemistry, though, I think about the chemistry diversity that's hidden in plants, because it's a critical functional trait that structures community interactions. And so instead of looking at this diverse tropical forest and see all these different kinds of species, I actually think about all the chemistry that could be in that system. Ecologists, however, in the past have focused on single compounds rather than the complex arsenal of defenses that plants have and that are encountered by insect herbivores. So this is just a subset of volatile compounds that are emitted from this wheatgrass. As you can see, there's a lot of different compounds, so there's richness diversity here, but there's also different classes of compounds, so there's functional diversity here, and there's also a lot of structural diversity here. Specifically, especially if you think about these aromatic rings versus some alkenes or alkenes or alkanes, there's a lot of different diversity just within this snapshot of volatiles from a single plant. And so the key here is that a plant, oh, I think I missed something, but okay, sorry, I missed something. So I just want to tell you though that even though we know that plants are comprised of a lot of different kinds of compounds, there are actually estimates of greater than 1 million unique phytochemicals, but only less than 10% of those compounds have been identified. And so when we really think about chemistry and how plants use that to interact with other organisms, a lot of times we're really looking at that small percentage. And there's got to be a reason for all these other compounds. And so my research really likes to think about it in an untargeted way or an approach. And I like to think about how I can actually still incorporate these unknown compounds even if I haven't been able to identify them. And so the newer paradigm, and this is really becoming more common, is that, yeah, there isn't just a single compound, or there isn't just one compound that's having an effect. There's actually this diversity that's driving plant-insect community interactions. And so there's been a lot of review papers. There's been experimental data papers by really famous chemical ecologists, and they all are agreeing on one common thing, that plant diversity is a functional trait. And so there's been this kind of hypothesis too, that's been developed, where if you think about, so this is a conceptual figure of a plant. Each of the colored shapes represent a unique compound. And so when we think about chemical diversity, we can think of high versus low categories of diversity. So in this case, this is an example of high chemical diversity. So there's the same number of compound richness, and they're evenly distributed. So there's an even abundance, right? And what we're finding in empirical studies is that the higher chemical diversity tends to have more herbivore species, but at lower abundance, resulting in low herbivory. And so the hypothesis for this talk, and what you'll see as I talk about chemical diversity, is that when I'm talking about high chemical diversity, I'm really thinking about a better defended plant. It tends to have lower herbivory. Conversely, here's the conceptual figure of what a low chemical diversity plant looks like. There's fewer richness, but what's really going on is that one unique compound is dominating the composition of that plant. And so what this is saying is that it's dominated by one or a few compounds. That means that there might be fewer herbivore species, but they're there at higher abundances. So there's more of them, and then they tend to result in higher herbivory. And so this hypothesis has really guided a lot of my work, and actually some of my work from my graduate career has actually helped shape this hypothesis. So briefly, I was looking at interest-specific phytochemical variation and how that regulated community interactions among specialist herbivores and their associated parasitoids. So this work took place on the eastern Andes of Ecuador, so in a cloud forest. And I was really, again, interested in interest-specific variation. So I was studying this plant, Piper kellii, and I was interested in looking at how the variation changed across populations. So I collected leaf material to do chemistry on across 32 populations. And then I also collected all the caterpillars I could find on this plant. And I just want to point out real quick, I know this looks like a little shrub, but this plant can grow into a mid-canopy tree. It has hundreds of leaves. These are very broad leaves. And so when I say I collected the caterpillars off of them, I would collect hundreds, maybe 500 per tree. And then we would rear these out. And so I know it looks like there's only there's only 7 caterpillars here, or 7 herbivores here, but these are species of caterpillars. So overall, it was the same genus, Eois, but 7 different species were feeding on the single host plant. That was pretty remarkable. So I reared them out to adult or parasitoid level, and I collected parasitoids across 4 families. So braconids, eulophids, ichneumonids, and tachinids. And so I was really looking at this community interaction driven by intraspecific chemistry. And so what I found was that phytochemical diversity reduced herbivore diversity and attracted more kinds of parasitoids. So this is a residual plot from a path analysis. I didn't put the whole path up. I'm happy to talk about it more. But a residual, this is looking at the residuals from a path. So on the x-axis is phytochemical diversity. On the y-axis is herbivore species diversity. And I found a significant negative relationship. So plants that had higher chemical diversity had fewer herbivore species. So right, that goes back to that hypothesis. But how did that affect parasitoids? I found, and again, these are the parasitoids reared out of those herbivores, right? I found the opposite effect. So as those same plants that had really high levels of chemical diversity had higher levels of parasitoid species diversity. So in this sense, this is saying that the high diversity, just in general, is indirectly drawing in more parasitoids to parasitize these herbivores. And so what that really looks like, just a small part of the path, it's a much bigger path analysis in the paper. But if we just look at this, it's like a double whammy. So plants with high phytochemical diversity had a direct negative effect on herbivore species diversity while simultaneously attracting more parasitoids, which had a negative effect on those parasitoids or on those herbivores. And so this chemistry is working both directly and indirectly. And so it kind of made me wonder at this stage, what's causing variation in phytochemistry? Is it genetic? Is it environmentally driven? But I want to point out that when I started working on this project, first of all, this plant species had never been identified. So you're working in the tropics with unidentified plants and insects, which it sounds kind of crazy, but it's true. And so I wanted to know the chemistry, but no one knew anything about the chemistry. So I decided that I was going to learn chemistry. And I was part of the group that helped characterize novel compounds produced by this plant. And I'm really proud. There's a common prenylate benzoic acid with a common name andrionic acid after me. And we found that this precursor had an enzymatic relationship to create a chromine. What's interesting about the chromine is that it happens to be photoactive, is what we found out. So if you shine light on it, it catalyzes this chromine and turns it into a dimeric chromane. And so this is a light-activated compound. The reason I looked at these specific compounds is because they were the most dominant, they were the most abundant or dominant in all of my leaf samples. So that's why I kind of zeroed in on these compounds. There's a lot more to the chemical profile of this plant. Anyway, so I'm noticing that there's changes to this chemical pathway due to light activation. And so I had a hypothesis that light availability is actually mediating the variation in phytochemical diversity, which then is cascading up to higher trophic levels. And so for this, I did an experimental test in Ecuador in the same cloud forest. And what I did is I took clonal plants of Piper kellii. I established them for roughly like 6 months to a year. And then we grew them in pots. And I put them in pots and I placed them paired clonal plants in the canopy. So I had one in the higher part of the canopy and one in the lower understory. So the higher canopy is exposed to more light, and then the understory is very shaded. It's a very dark shaded understory. So I manipulated light that way, but I also manipulated light using an elevational gradient. And so I wanted to look at how does differences in light availability affect the phytochemical variation. And so on the x-axis here is my measure of light availability. I use percent direct light transmittance. And then on the y-axis is phytochemical diversity. Again, these are higher, more defended plants. This is lower, less defended plants. And what I found really surprised me. I actually found that plants that were in the shaded forest understory had the highest levels of phytochemical diversity compared to plants that were in those gaps, or the gaps in the understory or in the canopy. I found that they had actually lower phytochemical diversity. So plants in the sun had lower phytochemical diversity, and plants in the shade had higher phytochemical diversity. This was interesting. I think, my thinking is, I still would have to tease this apart more, But I'm thinking because these compounds are photoactive, or the dominant ones are photoactive, when the plant is exposed to sun, it causes that pathway to it just turns that pathway on. So the plant is kind of canalizing and only making those compounds from that pathway. Whereas if you have plants in the shade, maybe the photoactive compounds are being made, but then there's a bunch of other pathways being made by that precursor. So that same precursor is working multiple pathways. But the sun kind of actually decides what pathway you're going to focus on, and this is the dominant compound coming out of it. And so that's my hypothesis why we're seeing this effect. But the big question is, okay, so light's is definitely affecting the phytochemical diversity. How is that affecting the herbivore community? And so on the x-axis again is phytochemical diversity, high diversity, low, and then the y-axis is herbivore damage. And I found that plants that had the highest herbivore damage correlated with really low phytochemical diversity. So the low chemical diversity is the poorly defended plants. Whereas plants that had super high phytochemical diversity, again, they were in the shade, they had the lowest herbivory. So in this case, again, we're also seeing that high levels of phytochemical diversity, even if it's driven by an environmental stressor such as light or an abiotic factor, would produce, has an effect on herbivory that cascades. Okay. So overall, from my work, from my dissertation work, it was a really important learning opportunity for me to think about how intraspecific phytochemical variation can cascade across the communities. I found that lower light levels increased phytochemical diversity, which had a negative effect on both herbivore species diversity and herbivory. And at the same time, I'm still seeing this dramatic indirect effect where the plant is bringing in more parasitoids that again regulate those same herbivores. And so I just wanted to point out that the phytochemical landscape is complex, and that I would suggest that trophic interactions are contingent upon this variation. So this variation across the landscape is really what's causing these interactions. And so this work really drives my passion for diversity and has built the foundation for my current research program. So my overall question in my research program is, to what degree does phytochemical variation inform ecology, pest management, and biological invasions? And I really come at it at the intersection of community ecology, plant insect ecology, and chemical ecology. And for my talk outline, I'm going to talk about two research, these are research projects that I conducted as a postdoc. They're very different systems, but some of the outcomes of these research projects are where I'm at today for my current faculty position. So I'm very new, I'm starting a research program, and so these are kind of the two main directions that I'm going with, based on some work that I did as a postdoc. I didn't say that very well, but I think you all get it. So for the first part, I'm gonna look at a trait-based approach to testing the influence of intraspecific phytochemical variation on herbivores. And then for part two, an untargeted metabolomic approach to the ecology and management of an invasive species. And then I'll end by reiterating the two of the big themes of research going on in my lab at Michigan State University. So for this one, this is when I was doing a postdoc at Michigan State University with Will Wetzel. I received a USDA NIFA postdoc fellowship for this work. And this is really thinking about chemistry as a trait-based approach and thinking about how we can manipulate certain neighborhoods based on chemical traits. I want to point out that I got to work with really awesome undergrad students and got to mentor them, and a lot of this work couldn't be done without them. And so I love working with students, so keep me in mind if you're looking for graduate programs. So the background to this. So intraspecific diversity cascades up to higher trophic levels as genetic diversity of the plant increases. So this is a conceptual figure of a goldenrod, and the colors, the different colors indicate unique genotypes. So there's a seminal paper by Kretzinger et al. Where they found that the number of plant genotypes, as that increases, it causes primary productivity to increase. Similarly, it also increases herbivore richness and predator richness. And so these results suggest that diversity begets diversity. So if you have higher diversity in your primary plant producers, you're going to see that cascade up trophic levels. And the idea here is really getting it up to these predators, because the more predators you have, the better control you'll have on your herbivores. So in a separate situation, this is an example of what a monoculture looks like in typical agroecosystems. So in this case, we don't have unique genotypes. We actually tend to have the same genotype. And that's usually for purposes for farmers to get higher yields of a specific crop, vegetable or fruit. But however, the problem here is that these dense stands of the same genotype become very apparent to herbivores, right? So it becomes very obvious where their host plant is. It's kind of like a big sign, like saying, hey, I'm right here, I'm so big, you can't miss me. And that's really one of the main reasons why we see a lot of insect outbreaks in agriculture. And then on top of that, we have to manage it with pesticides, whatnot. But that is a big problem. So a solution that is becoming more relevant in integrated pest management is increasing intraspecific genetic diversity in these systems. However, even though we're increasing genotype diversity, we're not always finding the same predicted effects that Kretzinger found. We're not always finding decreases in herbivory because we're getting higher levels of parasitoids. It's really unpredictable. Why is that? Well, I would argue that we're not taking a trait-based approach to this because arthropods, insects, are interacting with plant traits, not genetic diversity per se. And so if we're not selecting for genetic diversity with the right traits, then we might be missing it. And so my research wanted to identify what were the important plant traits that mattered. And shocker, I'm going to think about chemistry. And so one key trait that I wanted to think about was turnover in chemistry. How different are you from your neighbor? And so in this system, I really looked at the measure of beta diversity. And so for a really basic definition, phytochemical beta diversity is the chemical turnover or difference between neighboring plants. And so for this experiment, I want to maximize that difference. So my hypothesis is I'm testing associational resistance hypothesis, thinking about your neighbor, it being different from your neighbor. So does having high dissimilarity of phytochemical diversity compared to your neighbors, does that increase your resistance to herbivores? And so in this case, instead of the visual colors being different genotypes, I'm actually indicating that the colors mean they have different chemical profiles. So you want to be different. Okay, so how did I test this? So I used these integration lines, these tomato integration lines. I'm going to go over what those are here in a second. And I used those to experimentally manipulate phytochemical diversity. My model system was Solanum pinelli integration lines. And this is a powerful system because the genetics have been very well characterized, but so has the chemistry. And so this was a really nice system to work with because I didn't have to do all the hard work of characterizing an unknown plant species. So I actually really like that aspect of it. So briefly, what integration lines are. So this is Solanum pinelli integration lines. This is our cultivar. You can think of it as like your Roma tomato you buy in the grocery store. Plant breeders back-crossed the cultivar with the wild type Solanum pinelli, which is endemic to Peru. And so this wild type has very different chemistry. It's almost like a succulent. It's a very different looking tomato plant. And so they bred it because they want to reintroduce genetic diversity and chemical diversity back into our cultivar systems. And so with these integration lines, it resulted in 76 lines where each of the lines were identical to the parental cultivar, but there's a single chromosomal segment that's been introgressed from the wild type into these chromosomes. And so again, all of the genetics have been identified, well, not all of it, but the genetics have been pretty well studied in these integration lines, and so has the chemistry phenotype. So out of all those lines, I selected 9 to quantify chemical dissimilarity and defensive traits relative to the cultivar. So I just want to point out, and I'll say it a couple times throughout the talk because it can get confusing. Whenever I talk about dissimilarity, I'm always talking about the biggest difference, and I'm always comparing my lines, which are in colored, to the cultivar, because everything's being compared to that parental plant. So when I chose my lines, I decided to focus on 3 big classes of defensive traits that are known in tomato. So these are some of these are specific to tomato. So terpenes, that's kind of broad, that's across all plants. I looked at plants that pretty much had all their terpenes knocked out. So there were hardly any terpenes compared to the cultivar. And then tomatoes have these really interesting thing called acyl sugars. They're little glandular chemicals that are on trichomes, and they are very sticky and they can inhibit feeding. They can be pretty unique to tomatoes, but I selected ones that had reduced acyl sugars compared to this cultivar. And then finally, I looked at these proteinase inhibitors. These are so when insects are feeding on plant tissue, they use this enzyme to break down the amino acids. Well, plants can contain this thing called a proteinase inhibitor, which actually blocks the insect's enzyme from being able to break down the amino acids, and essentially it kind of starves those insects when they're feeding on it. And then how I calculated this is I calculated chemical dissimilarity using the Bray-Curtis distance index. If you want to talk more about these methods, I'm happy to talk to you about how I did this. But really, the big idea I want you to take from this is that I looked at these different traits, defensive traits, and I maximized the difference. So whenever I'm talking about it, it's like these have their terpenes knocked out, these have like their acyl sugars knocked out, and these have their proteinase inhibitors knocked out. Hopefully that makes sense. Okay, so now, I'm just saying these are some of these are specific to tomatoes. So if you want to do this with your system, I would suggest focusing on defensive traits that are important for your system. Okay, so for the experimental design, I want to give a big shout out to Kellogg Biological Station. So I did this when I was a postdoc, and I really loved working at Kellogg Biological Station. It was also really nice because I wanted to test the system in the field. I wanted to see natural recruitment of insects. So my design was this. So I had neighborhoods that had 4 plants. The focal plant is what I collected all the data off of, like all the insect data that you're going to see. But what I manipulated was the neighbor. So all neighborhoods had the same cultivar, and it's the neighbor that changed. So either the neighbor was the same cultivar, or the neighbor had those terpenes that were really reduced, or the neighbor had reduced acyl sugars, or the neighbor had reduced proteinase inhibitors. For purposes of this talk, because there's a lot of data, I'm going to really focus on terpenes because this actually was the class that I had the biggest effects with. And we'll talk more about that in a second. So not only did I manipulate chemical diversity, but I also manipulated the number of genotypes. So thinking kind of about genetic diversity. So I either had monocultures of 1 genotype, dicultures of 2 genotypes, or polycultures of 4. And then there was, so again, we collected insects from all the focal plants, and I had 2 types of insect collections. One was a sticky trap, and I'm pointing this out because the sticky trap, and I'm not going to get too much into this on this talk, but you're getting a very different guild of insect, right, with the sticky traps. You're getting more of those flyers or those long distance dispersal insects, as opposed to the beat sheet method. I don't know if anyone's done the beat sheet method. It's as kind of bad as it sounds, where you like literally hit the plant with a stick and you collect everything under it in like a little tarp or blanket or a catching tray. But these were, I wanted to point out, like you can see these are kind of more of your crawler, less mobile, I guess, predators and herbivores. Okay, so the big question is which is it, genetic richness or phytochemical beta diversity that's driving community interactions? We'll start with genotypic richness. So on my x-axis are those different neighborhoods, either monocultures, dicultures, or polycultures. On my y-axis is insect abundance. I'm going to show you the predators with black data points and then herbivores with green data points. For this one, I'm going to just show them all at once because I did not find a difference in insect abundance within either predator or herbivore guild. So yeah, there's more predators in the system than there are herbivores, but if you look within each of the guilds, there's not a big difference depending on the number of genotypes in that neighborhood. So now for phytochemical beta diversity. So the y-axis is the same, but for my x-axis, I now have my cultivar monoculture, because everything's being compared right to that cultivar. I then have a diculture of sesquiterpenes, and that means that they're reduced. I should write reduced there, but that means they're pretty much knocked out. I think it was like 75 to 80% knocked out. And then I had a diculture of just monoterpenes, and they were also knocked out. And then I actually put all the terpenes in a polyculture together. Okay. So in this case, it's both knockouts of sesquiterpene and monoterpenes. Let's start with, I guess, the predator. I couldn't remember what it started with. So here's like the baseline for the predators in the control. In the reduced sesquiterpene diculture, I didn't find a significant difference in abundance. But when we looked at the reduced monoterpenes, I found almost a threefold reduction in parasitoid abundance just within those neighborhoods compared to that cultivar. And then for the terpene polyculture, again, I didn't find much of a significant difference. For the herbivores, here's my baseline for the monoculture. For the sesquiterpenes, not a significant difference, but I did see almost a twofold increase in herbivores in those same reduced monoterpene dicultures. But I didn't find a significant difference so much in the polycultures. So while there's not a lot of significant difference, what I did key in on was that the monoterpenes seemed to have the biggest effect with both my crawlers and my flyers, my flying insects. And they also had contrasting effects. I want to point out, I kept saying dicultures and polycultures. I also want to point out that it seemed interesting to me that this was only found in the diculture, but we couldn't see it. We couldn't see those reduced monoterpene effects in the polyculture. And that could be because you need a certain number of that chemical trait. Like you need a certain density within that neighborhood for it to have an effect. So maybe in these polycultures, the monoculture was kind of diluted out. The insects weren't really picking up on the reduced monoterpenes because there are monoterpenes in those reduced sesquiterpenes plants. Hopefully that made sense. So what this is telling me is that a specific chemical class matters and that the density of that trait in the neighborhood matters. And I think I said that. So what this all means for broader implications, what does this mean in the big scheme of pest management agroecosystems? I suggest that we should really think about traits. So insects are interacting with those phytochemical traits, not genetic diversity per se. And so when we think about intracrop diversity practices, we should really select them based on specific chemotypes. I think we'd go a longer way if we're really keying in on the traits that are being different. And so in this case, I'm really interested with terpenes. I think there's a lot to work with there, and you'll see that that's kind of where I'm going next in my research program. But ultimately, I want to figure out what the combination of chemical traits, specifically terpenes, are, and then I can work with plant breeders to engineer varieties so that I can manipulate chemical diversity into that monoculture, right? Because I think you really need to disrupt it so that it's harder for the insect pests to find. Okay, I think I'm having a good time. Okay, so this is my second part. So again, this is some work that I did. I got funded by a USDA APHIS grant, and this was a second postdoc I did. I did this at Louisiana State University with Dr. Jim Cronin. And we were trying to understand mechanisms of competition with native and invasive Phragmites australis using a untargeted metabolomic approach. Okay, before I get into Phragmites, does anyone know about Phragmites here in Michigan? It's everywhere, right? Is it a cosmopolitan grass? Who said that to me earlier that they read a paper? Anyway, side note. So there are dual roles of Phragmites australis. I want to acknowledge that before I get into this talk. So here in the Great Lakes area, it seems like nobody likes Phragmites australis because it's a very aggressive invasive plant. It reduces biodiversity of the native communities, and it also threatens shorelines. I'm going to explain why, but it kind of takes over and it's really hard to remove. However, in the Mississippi River Delta of Louisiana, Phragmites is a foundational species in this wetland. In fact, it's really important for stabilizing the coast, and it can build land. However, I don't know if you've read the news recently or thought about Louisiana, but Louisiana is going underwater. So a lot of these really important coastal areas are going underwater with sea level rise, erosion, all sorts of things. But this plant all of a sudden kind of started dying off, which exacerbated the land loss problem. So this is a picture of this recent dieback starting in 2016, and you can just see that there's not frag like this anymore. It's just these like mud flats, right, that eventually get washed out into the ocean. And so I wanted to look at how phytochemical differences were affecting between the native and invasive Phragmites australis, in hopes of maybe pinpointing what trait would be resilient and that we could use to restore these coastlines. So yes, I want to restore the coast with Phragmites australis. Here, you would not want that. So I wanted to kind of make that a key point. I also think it's important, though, to think about how we can manage to keep it alive, because I think I can use some of the knowledge that I gained here to help kind of slow its growth down here in the Great Lakes. So ultimately, I would like to help manage invasive Phragmites australis here in the Great Lakes area, as I'll talk more about. Okay, so I told you that Phragmites is really important for stabilizing the coastline, and this is the reason why. So this plant grows really tall, like 2 times taller than me, which is, if you can believe it, really tall. It has these dense stands where you can barely walk through it. They're just so densely packed. And then below the ground, that's where the problem is in the Great Lakes, but why we want it in Louisiana is they have this really dense rhizome root structure. So it's really thick. It shoots down about 1 meter into the soil. And if you know anything about vegetative growing grasses, if you cut the top off or try to do something to the top, it just sends more rhizome roots into the underground. And so it actually helps it grow. And so a lot of the problem is we try to cut it down, but we're really just helping it spread underground. Anyways, that's a side note. But in the Mississippi River Delta, there are 3 main lineages of Phragmites that, again, are the most dominant wetland grass in the area. There are 2 native. There's a Delta and a Gulf lineage. And then there's also the non-native European, which we have here in the Great Lakes. So we have the same invasive, but there's different natives here and there's different natives there. Alarmingly, we found that Phragmites australis has started to experience a widespread dieback starting in 2016. And so this is a picture, This is a picture of a stand in 2010. You can see how robust and healthy it is. And then this is the same stand in 2021. And now the Phragmites is completely gone. It's dead, and it's actually now being invaded by invasive taro. So another invasive plant is now coming in and succeeding. The problem, though, with this is that taro's root structure doesn't go 1 meter deep into the soil. It's got this really flimsy kind of root structure compared to Phragmites. And so probably within 6 months to a year, this is going to be washed away if something else doesn't come to help stabilize the soil. So this is the problem. That's dying off. As I told you, land loss is attributed to flooding, sea level rise, subsidence, and erosion, but it's compounded by this die-off. And we really started seeing huge effects of land loss after 2016 when we discovered this die-off, especially here in the Mississippi River Delta. So this is a projected 50-year flooding map. So purple and red are showing you where flooding is gonna, it's gonna be over 15 feet. And you can see that the whole coast is expected to increase in flooding. But if this plant is dying off, we're gonna lose even more of that soil. And I wanna say that there are estimates that Louisiana is losing a landmass equivalent to 30 football fields per day. It's like pretty crazy. So this is a huge problem. There's a lot of money going in to try to, especially down at the Delta, to try to keep that land there because it's so important for so many reasons. One of the main culprits of this die-off isn't necessarily the sea level rise flooding. It happens to be this exotic Naponaclerta biwacoensis scale. Scales are pretty nasty insects. I don't know if you know much about them, but you can imagine a stalk of Phragmites is taller than me. At each node, there can be hundreds of these scales. So there can be thousands of scales. Each scale, if it's a female, is housing hundreds of crawlers. And so when the crawlers get big enough, they emerge and they have 72 hours to move. So they have a 72-hour dispersal window until they find a suitable place to establish. And then the cycle starts over. What's also interesting, I'll say it here. So we've done a bunch of work. So I'm going to kind of combine different projects in this part of the talk. But we wanted to understand the density of this scale across the coast. So we looked at Mississippi River Delta, Atchafalaya Basin, Vermilion Bay, and Rockefeller. And as you can see here at the Mississippi River Delta, insect scale densities were 3 times greater at the Mississippi River Delta. This is where the die-off is the biggest problem. It's also where we believe the invasive scale originated. So it came through on cargo and shipping boats. The other really unique thing about this system, and this is true across lots of systems, is these closely related lineages. There's differences in susceptibility to the scale and thus to the die-off. So the invasive European is actually quite resistant. We're not really finding stands of invasive plants that are dying off. In fact, they remain pretty healthy. And I'll show you some data here soon. They're also they're more resistant to the scale. However, the native Gulf and the native Delta, they're extremely susceptible to the die-off. The Delta in particular, because the Delta is adapted to flooded areas, like more on the coast, whereas this native Gulf is found more in like roadside ditches. It doesn't handle flooding as well. So anyways, but the native Delta, this is the most susceptible. This is what's dying off. So we've done a lot of work trying to understand the differences in resistance. One thing we found, these are the different lineages. Here's the invasive and the 2 natives. The invasive, time and time again, tends to have fewer scale densities. So it does seem to defend better, or is not as suitable of a host as the native lineages are. Okay, so I want to take a trait-based approach to understanding competition and plant invasions. I don't have the answer today, but that's kind of a long-term goal that I have for my research program. And so for this part, I want to talk specifically about how plant volatiles are affecting scale densities. So when we did this survey across the coast, we collected rhizomes from both native and invasive lineages, and we started. We have like a source population for common garden experiments back in Louisiana State University. And this is an amazing undergrad student, Dakota Babin, and she was the primary person running this project. So we wanted to collect plant volatiles. And so we did this really nifty bag experiment where we had made these portable vacuum pumps where it pushes in clean air and it pulls out the volatiles. And we created a headspace by using the oven roasting bags. So that's how we collected them in the field. And then Dakota, she extracted all of our traps and ran them on the GCMS to get the volatiles. Okay. So I know I talked a lot about chemical diversity. Remember, higher chemical diversity, I'm hypothesizing, means a better defended plant. So is that true with VOCs? So I want to point out in this measure, this is a diversity. So that means it's compiled of, I think it's like 23 terpene compounds that I identified. So even though I'm showing you one number, there's a lot of terpene compounds that make up this number. So on the x-axis is volatile diversity. So this is low diversity, this is high diversity. And then on the y-axis is scale abundance, and this has been standardized to match average per meter of stem. So this is the scales per meter of stem. Orange is the native Delta. The triangle, blue triangle, is the invasive European. For scales, especially in the Delta lineage, we didn't have enough representation in the EU, but we did not find an association with terpene diversity and scale densities. So to me, I don't think scales are tracking terpenes. I've done some other stuff and I don't think they're tracking the terpenes. However, what was super exciting is that, again, we have the low diversity, high diversity, but on the y-axis this time is parasitoid density. And I found for the Delta plants that as plants that had higher terpene diversity had higher levels of parasitoid density. So the parasitoids are tracking those terpenes. And I want to point out that's what the literature says. And for me, this was like super cool because I'm like, oh, here's empirical data that really shows that in the literature. So that was a very satisfying result. I'm not surprised by the result. I was just really happy to see that it happened. I also want to point out, and I should have said this sooner, when we collected those rhizomes across the coast, we at the same time in the field that's when we took the scale and parasitoid measurements. And so then we established them in a common garden, and then we collected the volatiles about 6 months later. And so you're seeing a conserved, I'm thinking that this is more of like a conserved effect. Like the terpenes have evolved to draw in those parasitoids. So we're seeing this after they've been established under the same environmental conditions. So that was really cool. Oh, and last thing, parasitoids, as we know, are the only thing that can inhibit the scales. Scales are really hard to treat. They have a waxy coating over them. They're a huge problem in, I know, fruit trees, fruit crops, because you can't spray them with insecticide. That waxy cover is really hard to penetrate through. So really our best bet is biocontrol. So that was really cool to see that effect. So another experiment, we wanted to look at, I forget what this one was. Use the fully crossed experiment. Why is it not moving? Here we go. So we had 3 lineages, the 2 native, the invasive. We took soil from healthy and die-off stands, and we fully crossed that with the presence of scales or no scales. And then we collected volatiles, again, using that same method in the field. This is, again, a different experiment from what I just showed you. Okay, so I broke this down as a conceptual path diagram to show you so I'm looking just in this path, just die-off soils, its effects on scale abundance, and then I'm going to show you what we got for terpenes. So this is for the green represents the invasive plant. So we found in those die-off soils, and we're assuming that there's some problem with the microbes. We don't quite know. I would love to learn more about soil microbes and would love to collaborate on that. We found that with scale, when in the presence of scale, terpene diversity decreased 3%. It wasn't significant, but it wasn't upregulated by any means. However, with the 2 native plants, we found a much more dramatic decrease in that terpene diversity. So this is the Gulf one. There was a negative 16% reduction in terpene diversity. And for this native Delta lineage, we actually found a negative 26% in reduction of terpene diversity. So that's a huge reduction, especially in this native compared to the invasive. If the plants are really using those terpenes to bring in the parasitoids to control those herbivores, the European isn't really changing, but the Delta is having a huge negative effect. They're probably not bringing in as many parasitoids. So this one, though, is now looking at the healthy soils. So again, with the presence of scales, for the invasive, we found that terpene diversity in those healthy soils increased by 32%. So that's a huge upregulation of those terpenes. For the other natives, the Delta and the Gulf and the Delta, they also had increases in terpene diversity, but only about half as much as the invasive did, so about 14%. And so what's going on here is it does seem like that terpene diversity is higher in these invasive lineages in general, which may be the key to why they're doing better. And so that's all I'm going to show for my postdoc, but I'm really interested in thinking about trait differences, particularly in terpene volatiles, and how they're linked to resistance against the die-off, but also to insect pest invasion. And so I wanted to say I do plan to look at volatile traits of Phragmites australis in the Great Lakes region. So this is my lab. I'm super excited. I just started, like I said, about a year ago. I have a postdoc, a lab manager, and a PhD student. And my program is really thinking about linking insect community structure to both pest management and biological invasions. And so the first research direction that we're developing is, again, back to those agroecosystems. I really want to look at the mechanisms of odor complexity. And so, again, I found some really cool effects with terpenes, so I really want to focus on terpenes, and I'm thinking about it in terms of either a uniform odor plume, which kind of mimics those monocultures, versus a more complex odor plume. So in that case, I'm thinking about variation in odor diversity. And so my hypothesis here is that if it's a uniform odor plume, herbivores are going to have an easy time finding it because it's really reliable, it's really apparent, it's a strong, uniform odor that they can track consistently. Whereas the literature suggests that natural enemies have a better time navigating through complexity because they're constantly trying to understand reliable versus unreliable signals. And so they are better at dealing with a less predictable, complex landscape. And so in this case, I'm hypothesizing that natural enemies will locate hosts across trophic levels using indirect cues from the herbivore activity or herbivore-induced plant volatiles. And then for this direction, so that's in more of an agroecosystem. I'm thinking of doing more pest management, integrated pest management. That's kind of the goal there. But for this direction, I want to keep working with Phragmites, and I really want to build up that invasive native competition. I want to understand what mechanism is driving competition. And so for this, and I don't know why my picture is missing, but in the Great Lakes, there's also native and invasive Phragmites. I'm interested in understanding the chemical trait differences between the two in terms of competition, but I'm also interested in how does climatic gradients affect that competition? Does it enhance it? Does it dampen it? And does that exaggerate those chemical differences between native and invasives? So for this, my postdoc is kind of leading this project, but we're looking at, we've collected so far populations of native and invasive plants all along Lake Michigan, so all around the state, and we're creating source populations back at Michigan State greenhouses so that we can start conducting some competition experiments this summer in a common garden experiment. And so I'd love to talk about any of those topics if anybody wants to collaborate or think more about it. And so with that, and I only have 4 minutes, so I was under. With that, I want to thank all of the collaborators I've worked with, from University of Nevada, Reno to Michigan State University to Louisiana State. I've worked with a lot of awesome graduate students who are all listed here. I've also couldn't have done the work without all the undergraduate mentees I've had. It's been really fun teaching them science, but also seeing them get really excited about it. So that's a really fun aspect, I think, of this job. And I want to thank my funders, NSF and USDA. So thank you, and I'm excited to answer any questions. I'm like, I clapped because it was over. Thank you for your attention.
Okay, I think we have time for a couple of questions.
And I'll put that up if people want to see.
Thank you. Great talks, and
thank you.
Thank you. It's really interesting how the correlation between the plant's chemistry, herbivories, and parasitoid diversity, especially parasitoid diversity. So the one thing is usually for the parasitoid, especially on the parasitoid hymenopterans, they are usually host specific because due to long evolutionary history between the host and the parasite. So I was really curious at how the plant chemical diversity of the plant reduces the host, especially herbivory diversity, but increase the host diversity. So do you have any explanatory mechanisms or a specific trait that boosting the diversity of those parasitoids?
Yeah, that's a great question. So I will say for both projects, there are parasitoids that are both generalists and specialists. I think that will play a huge role if they're generalists, if they can track a lot of things. They're kind of, they're used to a lot of different. I don't know if they're necessarily tracking a specific chemical cue. The specialists likely are. I have not done any assays where I actually look at the difference. I haven't done any assays, I think, in at least the Piper system, where I looked at differences in plants that have had herbivory versus those that have no herbivory. So I don't know if there's novel compounds being produced based on induced defenses. I also I have looked at that and the Phragmites system. I'm not necessarily seeing new compounds being formed. I'm mostly seeing abundances changing. And I think the reason I'm not seeing really, and I've seen this in other chemical traits that I've looked at with intraspecific differences or intraspecific variation, I'm not really seeing novel compounds being produced. It seems to be affecting more of the evenness or the abundances of individual compounds. And I'm kind of thinking that might make sense because they're closely related. They're following similar biosynthetic pathways. And so I actually think that the ratios are the key difference that I would love to, that's what I'm trying to kind of disentangle. Yeah. Do you want this big microphone?
This one's good. Cool talk. I was curious about when you see Phragmites dieback, is it often linked to particular like abiotic perturbations, like intense rainfall or temperature? And if so, do you think it's mostly